Please join me in welcoming Dr. Kenneth Kendler, a world-renowned expert on the genetics of psychiatric and substance abuse disorders. Dr. Kendler is a highly cited researcher whose work has transformed our understanding of how genes and the environment contribute to conditions like alcohol use disorders, major depression, and schizophrenia. He employs key research methodologies, including large twin studies and molecular genetics, like genome-wide association studies, to unravel the genetic and environmental mysteries of these disorders. Dr. Kendler is one of the most often cited psychiatric researchers. He has published more than 1,300 articles in peer-reviewed journals. Dr. Kendler serves as the editor of Psychological Medicine, a leading international journal. He has received numerous awards and honors, including the International Society of Psychiatric Genetics Lifetime Achievement Award and the World Psychiatric Association's Gene Delay Prize, which honors psychiatrists who have made a major contribution to the discipline. Currently, Dr. Kendler serves as the director of VCU's Institute for Psychiatric and Behavioral Genetics, where his goal as a teacher is to inspire the next generation of researchers to appreciate the complexity of human behavior. Without further ado, Dr. Kendler.
Thank you for that introduction. So I have some ambitions for the talk tonight. We've got almost 70 minutes. I want to try to take you through the history of my discipline, to give you a background about how we began asking individual questions, why disorders ran in families, how they did that, and I want to bring you up to the absolute cutting edge. We're going to be talking some about the technical aspects of the human genome. I have not brought slides. I'm going to try to give you the major conceptual points. Also, I get bored in hearing myself talk because I know my speech that's coming out. So an interaction with you, I certainly have built that in. And if there are points in which I'm really losing you, I would prefer you raise your hand because you're probably not the only one. And we have enough time to have a good back and forth here. Okay. So at the end, what I want to show you is important discoveries that we have made in this field. I mean, transformational compared to where we would have been 20 years ago. Much of this driven through the extraordinary technical advancements in the world of molecular genetics. And try to then show you where are we and how are we struggling with taking those advances in the basic underlying risks for psychiatric disorders and trying to translate them into a biological understanding that then could lead to pharmacologic targets so that eventually we can take these insights and bring them back into the clinic with new, effective, empirically developed treatments. That's the goal of all of us in this field. Okay. But it's been a long tale, which I will start really very much in the beginning. Psychiatry as a discipline began shortly after the French Revolution, the first time that physicians began to look at and care for what would have been called mad folks then. In those days, we were called, if you were in England, you'd be called a mad doctor, and if you were in France, you'd be an alienist. Psychiatry is actually a term that only developed in the early 20th century. And that was the first time when psychiatric patients began to be clustered into what we would then call asylums, large numbers. And a group of physicians began thinking that they should specialize in trying to care for those individuals. And in the early part of the 19th century, you had foundations that became the American Psychiatric Association in England and France, and they began to write textbooks. So we see this proliferation of this specialties, and I spent some years, 4 or 5 years ago, reading through many of those texts, these early descriptions, mostly because I wanted to understand how did they think about the genetics of the people they saw. Now, this is before Darwin, before Mendel. They had no concept of what genetics was, but it was clear reading through those books that they emphasized 3 things that I want to convey to you. First, almost all of those textbooks had a chapter about the etiology of madness or insanity. And in almost every single one of them, they said, well, it's quite clear from the patients we see that genetics is the most important cause. So it didn't take modern fancy research, and I'll describe that in some detail, to discover the importance of how much heredity influenced the risk for psychiatric or mental disorders. And you might think that this was only recognized in the field, but that's not true. If you go back to the very beginning of medical genetics, as doctors began to look all across disorders, which were the most heritable. And a major dominant early textbook in medical genetics in the 1830s began with the two most heritable disorders that he knew, and one was gout, which runs very strongly in families, and the other was insanity. So it's not a new idea in our field that hereditary factors are important. Now, what are the other things that they found in this situation? One is that they were puzzled when you actually looked at pedigrees containing members who were mentally ill. That you might have a pair of healthy parents, and they have an ill child, and then they're not ill, but an uncle is ill, or a second cousin, or a parent. It seemed that the disorders seemed to skip generations, and they had situations where parents were clearly ill and had 8 children, and only 1 or 2 of them were affected, and they pondered what could go on. You may know the term heredity comes from the term inheritance, so we inherit our land as we do our characters from our relatives. And they figured out, and they used a variety of different terms, predisposition, vulnerability. But by the late 1880s, when they knew almost nothing about the substance, they said heredity is important, but it's not the disorder that itself is inherited. What's inherited is somehow the vulnerability to it. And that must interact in some ways with extra experiences. So how is it that some siblings would be quite well and others ill? There must be something that is provoking those conditions. And that's all exactly right. We now know 150 years later. And the third thing they noticed was that if you look within families, you seem to find some families with the same disorder runs very consistently. Let's say melancholia or mania, or what somebody we would now call a psychotic illness, schizophrenia. But other families weren't like that they had quite different disorders in the different members of the families. And so they couldn't quite figure out, do these disorders run true within families, or there's actually quite a bit of variation? And that, by the way, is still a topic that we're struggling with today. So they got it pretty right without our fancy statistics, without our molecular genetics of these views. Okay. Now, the journey that I want to start on is a series of questions that we ask in the discipline of psychiatric genetics. The first being, do disorders run in families? That's kind of the basic feature that we want to understand. And how do you end up doing that? Well, I will describe to you a family study, one of the first that I did when I was done with my training. This was done in Ireland. It was a county in the western part of Ireland called Roscommon. If any of you have traveled, mostly the tourists skip Roscommon, by the way, but it's on the way to Galway. It's in the Connacht, the west area. And I started there because the Irish government had established a case register examining all the individuals who'd ever sought treatment. So I had a really good quality general sample there. We got NIH funding, and we hired a team of mostly social workers and psychiatrists. We had visiting nurses in the hospital who cooperated with us and went out to families. We didn't have any computing systems then, and they basically had everybody draw pedigrees of themselves, who their siblings were, their parents, and their children. We collected those cards and addresses. I brought the 15 people who were working as interviewers, and we spent 2 weeks being trained in how to conduct the standard interview. And then we basically randomized those relatives. So the interviewers who drove out all over the areas in Roscommon and Galway and Sligo and interviewed families in their homes, they didn't know who they were interviewing. Was it a relative of a control patient? Was it a relative of someone that had a mood disorder? Was it a relative of someone with schizophrenia? It took us about 4 years. We interviewed about 1,800 people. What we found is that if you went into a home in the west of Ireland, the trouble was not getting in, but getting out. So I would have done about 140 of those interviews, and an American doctor, oh my goodness, they sent their kids down to the local store and brought things back. They got out their family catalogs, and did I know their Uncle Kelly, who lived in Michigan, and it was a big deal there. So the cooperation rate in rural Ireland was like 97%. Rural Ireland at that point was a place where if your car broke down, the first person who drove by just stopped and fixed it. It's sort of that kind of rural culture. So we had very complete group of relatives. And we then we had people going out into the psychiatric hospitals, identifying the relatives, doing case summaries, and those all got put in little files. And then I and a colleague, it took us about 6 months, took each file one at a time, we didn't know who they were related to, and did a complex scoring of all the kinds of disorders we had. And what did we find in the end? And this will be the introduction to the basic results we know, how strongly do the major psychiatric disorders run in families. And the way we typically think about that is if you are a sibling or a child of someone with a disorder, how elevated is your risk to a risk of a person just like you in every other characteristic, but they don't have a close relative with mental illness. Example was in that study for the relatives of individuals who had schizophrenia, the rates in the control sample was about 0.5%, and the rate in the siblings and parents of the schizophrenics was about 6%. So roughly what that meant was you have about a 10-fold increased risk of schizophrenia if you come from relatives, because that's one way to see it, but 6% is actually a pretty low figure. And I would have spent time in my life doing genetic counseling. So a young woman comes in, has a brother or a father, let's say with schizophrenia, and she says, she's 30, let's say, or 35, she's mostly through a risk. I want to have children, what's the risk that they have schizophrenia? In fact, you go to these older family studies, and now you know, and the answer to that is it's like 2%. It's really quite low. It's above the general population, but fairly low, and most people are quite encouraged about that. Now, depression, it's a much more common disease, and it's about 3-fold increased risk in relatives. So the average rate of depression in the population is almost 15%. It's quite high. And if you have a close relative, sibling, parent, you have a risk of about 45%. So almost 1 in 2 of the close relatives of depression will develop a disorder in their life. Bipolar illness is more like schizophrenia. It's rare and carries about a 10-fold increased risk. So that's foundational information that really allows us to go forward in the field. We know that these disorders run in families. The next obvious question is why? So we think about some human characteristics like the religion you ascribe to or the language you speak that runs very strongly in families, and yet we know has absolutely nothing to do with your genetic effects. And political attitudes typically run in families, although not always. We all know situations where parents and kids don't agree, but it still tends to run on average. So there are clearly human characteristics that run strongly in families that have nothing to do with genes. So although there have been researchers in my field who want to assume, it runs in families, it has to be genetic, that's false. And so we've used 2 methods, and I'm going to skip over this fairly quickly here, that try to disentangle. We sometimes call this the nature-nurture problem, right? That if you're like your parent, is it because of the genes your mom and dad gave you, or is it because they raised you and exposed you to social attitudes and you went to church or synagogue, others with them, and all the cultural experiences would go along with that. So the twin method that we use, and this is something that really brought me to MCV, is simply this fantastic natural experiment of pairs of identical twins. And this occurs when a mother ovulates, and in the first 14 days of gestation, the zygote splits in 2. We still don't understand the science, what happens. And the father, usually in a single act of sexual intercourse, 2 sperms are, the sperms are racing out, and who gets to the egg first? And there are 2 sperms, each in one of those 2 eggs, and it produces individuals who are then genetically identical. So when they are born, identical twins share all their genes in common, and their environment accumulates differently. Dizygotic twins arise when a mother misfires and she ovulates 2 separate eggs. And those 2 eggs are fertilized by 2 different sperm, and they're just like brother and sister, but the same age. This actually was introduced in psychiatry in the 1920s. The first studies ever done, very small. And when we got here, one of the things that we wanted to do, the research team that I was working with here, is to try to do the first experiment where instead of studying only twins who were hospitalized and had severe illness, to study twins in the general population, which we did through the Virginia Twin Registry. And over a period of about 15 years, we actually interviewed almost 12,000 twins and their parents. So it was a lot of time, a lot of miles driving out into the countryside, interviewing twins. And in that way, we get a much more accurate picture because they don't have to present for a care to a physician or to a psychiatrist. So we were able to learn more about the common proportions. And using twin methods, I won't go into the statistics here, but I can if you're interested, you can actually carve about the proportion of the twin resemblance, that is, to the genes themselves, or the shared environment that they had and they grew up in. And what we found is disorders like major depression, anxiety disorders, drug abuse, run in families 85 or 90% because of the genes that are shared. The environment is important in some instances for depression, particularly for substance abuse, as you might imagine, but we have now quite a bit of evidence that the overwhelming reason why psychiatric disorders run in families is because of genes. Well, the environment will sometimes be important, and even if, in a few instances, critical. The other technique we use is adoption studies. So think for a moment about an adoptee who was adopted away at a young age and then was raised by a different family. That person then has a set of biological relatives who they've typically never met and a series of people who they're raised with, their adoptive family. And so that's another perfect natural experiment to ask, and I was involved early in my career in the analysis of one of the great studies that I didn't have any role in doing, but reanalyzing that Seymour Ketty did in Denmark, and he established a group first of 35 and then 70 individuals who were adopted away at a young age and grew up never having contact with their biological parents. And what it turned out is that when you interviewed their relatives, the only group of relatives that had a high increased risk of schizophrenia and schizophrenia-like illnesses were the biological relatives of the adoptees. It wasn't their adoptive relatives, and it wasn't the biological relatives of the controls. So this was really, by the time we got to the mid-1980s, the field had pretty well established that most of the reason why these disorders run in families is for genetic purposes. Now, that's an important finding. It involves in counseling. You can use it in very important ways to understand differences among different psychiatric disorders, but it doesn't give you a biological link to try to understand the etiology of the disorder. Now, that has been a goal for psychiatry for more than 150 years. The number of brains that have been dissected, the gallons and gallons and gallons of urine that has been collected, the number of times we've done spinal taps to look at the cerebrospinal fluid to try to understand what is systematically different in people with schizophrenia, depression, bipolar illness, and controls. And the fact is that almost all of that work has not produced meaningful findings. The buckets and buckets of urine, they found some difference, and it turned out that it was a result of the particular kind of tea that the hospital served. So it's just a series of these dead ends that occur. These are sort of become famous stories over time. I think we understand why, and I will take a little bit of detour. So let's think for a moment about our brain. These estimates vary, but the latest estimate is 86 trillion neurons that we have in our brain. And each one of those are connected up with 300 or 400 others. Neurologists have the brain kind of science they do, they have it relatively easy. In that if you have a major problem in a part of your brain that's involved in motion, right? My ability to move, my ability to walk, my ability to touch and be coordinated, or to be blind, or to become deaf, those parts of the brain we understand pretty well. They're the brain receives information from the environment around. It gives us our consciousness, your ability to understand my voice. And it controls how we walk, and our limbs are coordinated. But that's not where psychiatric illness lies. That's the easy part of the brain. Psychiatric illness involves things like control of your mood. We all have the experience of our mood going up and down. Things make us happy. We see our loved ones. We watch a funny movie. We drink a cup of coffee, we have a piece of chocolate, we watch the sunset. It's different among all of us. And parts of our pleasure centers kind of light up. What goes wrong with those when instead of those happening, that part of the pleasure, we call it hedonia, this capacity to experience pleasure, disappears? It's a frightening experience. You get up in the morning, you look outside, there's a sunset, you don't feel anything. You go and you hug your wife and she kisses you. You don't feel anything. You visit your grandchildren, that is the joy of your life, and they run into your arms, and you don't feel anything. That's what a severe depression is like, where it's as if, and people say, of course, this isn't my world. I don't understand. I can't really live like this. Everything is gray and dark. How do we begin to understand what the abnormalities of those subtle changes, small groups, probably thousands of neurons only, in the brain might be doing? It's extremely challenging. Psychosis is even subtler. So the abnormalities of psychosis are a capacity to accurately perceive the world around us, to be able to distinguish our thoughts inside our head from what you're seeing outside. So all of you now, while I'm talking, you're having some reaction. This is a great talk. It's boring. What am I going to do afterwards? We have a little thing that's running in the back of our minds. And having a healthy brain, we don't have any trouble distinguishing our inner thoughts and outer thoughts, we don't get mixed up. That's not true in schizophrenia. That is, one of the symptoms of schizophrenia is this impression that thoughts are being imposed on your mind. Someone is taking control of your will. And we now know that that's a fundamental confusion between the internal dialogue that we have and the outside world. Hallucinations are the same thing. Hallucinations are inner speech, but that's not what it feels like to them. And the capacity to accurately perceive reality. So I'll make a joke of it. You're a foreigner and you go to New York City and you say, "Doctor, all these yellow cars, they're following me around everywhere. I will see them." Of course, that's the cab rides. But that's not what it seems like to the person. By seeing those, that has some special meaning to them. They know they're being followed. So this is a long-winded way of saying that the standard approaches, we look at things like urine, which bathes all 87 billion neurons, trying to extract from that makes no sense. We have to somehow get to the specificities of what these disorders are doing. And what has been the major advances that I'm going to spend my time talking about is what genetic tools have we been able to get. And the metaphor I want to use is, if psychiatric disorders are kind of like a mountain, and the pinnacle of the mountain is the secret of what the biology is actually doing. What these little findings are like pieces of rope that hang down from the mountain, and we have to kind of grab them and work our way up from abnormalities in the DNA, which I'll be talking about in quite some time, to understand what those abnormalities at the DNA level are doing in the brain so we can intervene. And we've made some exciting advances. So now let me go back a few steps. A big event in psychiatric genetics happened in 1983. Jim Guzella, still working in a lab in Massachusetts General Hospital, found linkage to the neurological disorder of Huntington's chorea. It's a disorder that presents in the early 40s or 50s. It has very strong writhing movements that are uncontrollable. It's really quite odd to go to a Huntington's chorea clinic. People are moving around like that involuntarily. It is what we call a Mendelian dominant disorder. And that means if you have the bad gene, you always get ill. And it runs through families in that perfect way that you would have seen in your biology textbooks in high school, that great-grandfather to son to daughter to son, every generation has a 50% chance of getting ill, and you have enough kids, it'll get transmitted. That's the simple kinds of genetics that Mendel discovered. I was studying in Birmingham, England, quantitative genetics. It was published in Nature, and I remember quite distinctly picking up that journal and looking through it and understanding the impact that this is the first time we had used this modern molecular tools to actually identify places on the genome that we could track to follow. And I thought to myself, could we ever do something like this for psychiatric illness? Now, it so happened that 4 years later, I was in cars driving around Ireland. Ireland, the Irish Catholic Church is a great thing for geneticists. They have really big families, so that many of the people who we were interviewing in Ireland in that first Roscommon family study I told you about had 37 first cousins. Right? That just was fairly typical, and the SIB-ships were very large. And I was able to get funding now to try to apply this new technology of linkage analysis. We ended up spending 4 years. We had teams going. We had a team in Northern Ireland, and this was the first time that we'd gotten a collaborative arrangement between Northern Ireland and the Republic, and that was a little political dicey at moments. I have some stories to tell. I won't tell you about that. About a few times when that collaboration almost came apart. And they would go to hospitals, and they would sit down and talk with often the psychiatric nurses and doctors, and we'd hear this, "Oh, let me tell you about the O'Shaughnessy's. I remember the grandfather, and it was on Christmas Day that he drove his tractor into town screaming that he was God. And then the son," and they sort of reel out these pedigrees, which of course is exactly what we wanted, these very unique samples. And we came back with the largest collection of schizophrenia pedigrees, almost 300 of them from throughout the island of Ireland. We knew when we started that this method would work if we found genetic variants that had a big effect on the illness. Now, let me take a step back for technology purpose. First, human genome, 3.1 billion base pairs, roughly, right? It would sit on a hard drive. And it has an alphabet, which has just got 4 letters in it, A, T, C, and G for the 4 base pairs that you learned about. Those are the ones that are stacked in this double helix. We know a lot about the human genome now. It is, of course, organized into chromosomes, but within those chromosomes, it comes in little packets, we call those haplotypes, that are so small, about 15,000 base pairs for people of European ancestry, that it's never been broken up by a recombination. By recombination, every time, for the women in the audience, when your ovaries make an egg, and men in your testicles, they make a sperm, you have to go from 2 chromosomes down to 1. And you cut them in various, and then you stitch them back together, and that becomes the sperm and the egg that unites together, and then you start back to 2 chromosomes for your baby. That they're so small they've never been broken up. So that is a hunk of DNA that we can follow. The linkage analyses were eventually not successful. And they weren't successful because, and we now know this, that the size of the genetic influences for psychiatric disorders are not large. Some of you may be familiar with the BRCA gene. So BRCA increases risk of breast cancer in women. If you carry that gene about 70% of the time, and you live long enough, you'll get breast cancer. And for BRCA2, you'll also get ovarian cancer in that way. Another gene is ApoE4. That increased risk for Alzheimer's disease about 4-fold. So if we'd found genes for psychiatric disorders that had that much clinical effect, linkage would have worked. And it didn't, and now we explain why. Okay, so how are we doing for time? All right, good. The next major development was a technique that is called genome-wide association studies. And this was based on the next great development after the 1983 paper, and that's 2003, the human genome was sequenced for the first time. So that meant that you literally take a single human being initially and line it up from the very top of chromosome 1 all the way through the bottom of chromosome 23, and we knew how all the letters, it's actually about 98%. It took a bunch of years to fill in that 2%, but that's a small technical point. And then the question was, well, now that we have one sequence, how variable are those across human beings? Turns out that we took people in our audience, most probably we would differ, about 1 out of every 800 of those would differ between us, which if you do the math means that we differ in tens of thousands of those variants from one another. Very large numbers, even though we're otherwise quite alike. So as we began that sequencing, we were developing a technology of being able to measure those variants relatively inexpensively. If you have a base pair in your DNA that is variable, the technical term in genetics for something that's variable is it's polymorphic. It comes in more than one group. So we call it single nucleotide polymorphism, which becomes the word SNP. So if you want to be au courant in the language of molecular genetics, you talk about SNPs. And then when we measure them on SNP chips, right? Nice little line. And the SNP chips, when we started, had only 10,000 SNPs, and then 100,000, and then 1 million, and then 2 million. So the technology has grown up and up and up. So the field was very excited about these developments. There was the Wellcome Trust, a big English charity, funded a pilot study that was published in 2007. I remember very distinctly sitting in my backyard reading it. It was like a trial run. And they looked at type 1 diabetes, juvenile onset, adult onset. They looked at coronary artery disease. They looked at Crohn's disease. And they looked at bipolar illness. And they included about 3,000 people, which we thought then was an immense signal. And they sort of graphed that out. Where did they find those SNPs that differed between the cases and the controls? This is a much more refined method than linkage. If we think in a metaphor, if our genome is the state of Virginia, linkage analysis would get you into maybe a medium-sized city, maybe 100 miles around. While if you found a SNP marker using these genome-wide association, like in a block, that is the levels and amount of DNA there was much, much smaller. So the psychiatric genetics field kind of began to wake up. How are we going to try to use this new technology? Could we apply it? How many people would we have to study? Because we didn't know the effect sizes. Tiny effects take bigger numbers to study. I got a grant. I was going to study 1,000 cases of schizophrenia, and that's no small feat. I was on Aer Lingus, as I took a lot that time. I'm going to nail it. Right? Not even close. Okay. got 1,000 people, we matched them up. We didn't find anything. The field got 3,000. We didn't find anything. We got to 7,000. We didn't find anything. And at this point, the NIH, the National Institute of Health, who'd spent quite a bit of money on this, were going, I don't know, guys, we might not be funding any more grants. This is a whole lot of money going down the drain. And we said, just be patient. We don't know how small these effects are. And then a paper came out with 1 hit, and then 3 hits. And then I will tell you the story. I was privileged to be in the group that was collaborating and getting all the groups together to collaborate. Let me do a side story. I think we have time. The standard approach to academics is that you are rewarded when you find an important finding. You try to go to the highest mountain, plant your flag down and scream, I discovered this, I have publication priority, it came first to me. And then you get tenure and you get awards and people fuss over you, and that's sort of what we're supposed to do. The field was in this for 3 or 4 years. We each had our smaller studies, and we sat around a dinner table one night and say, you know, guys, this isn't working. We have to pool our data together. Because if we're going to be competing with them, we're all going to be publishing these small amounts. If we look across all of us and we combine our data, maybe we're going to find something. And then we have to say, but oh my God, we've got to find a common computing environment, and we've got to get a common data analysis pool. And that was the founding of the Psychiatric Genomics Consortium, which exists to this day. And I co-chair one of those groups, and I've been sitting on the schizophrenia one now for 15 years. And it makes me proud. That is, you know, we argue over who becomes first and last on these papers. Have like 400 authors. You know, it's kind of like the things you see from CERN. And sometimes your name is in the middle, and you've got to get a magnifying glass. Oh, you see, I'm right there. That's me. But the findings have really begun to flow. So I'd been on this call knowing that we had a finding that we found actually over 100 genome-wide significant loci. I was not able to go. This was announced and was going to be presented at the World Congress of Psychiatric Genetics. I wasn't there, but I heard the story, so I knew. And Stefan Ripke, who was the lead analyst there, stepped up, talked about it, and then showed the slide. And by the way, I've got a T-shirt of that slide. I almost thought I should wear it at this point. And it says, Schizophrenia Genetics, Progress at Last. And the audience just rose up, right? There were people in the back that were crying. They've been working for years and years, and something broke. The sample size got big enough. The amount came out. So we now have 400 of these variants. I can give you a slide, you know, how much this is going up. But the point is, we now have many of these little tag pieces of molecular DNA that contains a risk variant somewhere. We don't know precisely where. Remember this rope analogy. And how the heck do we get from there up to the underlying cause? That's been the major dilemma now. So let me now move more frequently to more recent events. There's an even more detailed approach to identifying genetic variants. 3% of our genome, a small proportion, is actually made into the proteins that run our body, that do all our metabolism, that build our bones, that get our neurons to work, that pump neurotransmitters backward and forward. The rest of it is all control variables. And we actually now have the capacity for sequencing those areas. They're called the exome because that's the part of the DNA that comes out. And unlike what we find from the whole genome sequence, we're figuring out which is the biologically meaningful variant, which is hard. The exome, we know precisely what it is. And when you get a variant in an exome, with our capacity now, we can take that little bit of exome, cut it out of a human being from their DNA, and put it into a mouse of various breeds. We can cut it out of them and put it into a monkey, into a marmoset monkey or a rhesus monkey. We can take that, put that DNA in a dish, take a bit of your cells that are taken from your skin, add some chemicals to de-differentiate them back to being basically like a human egg, and then cause them to redevelop into neurons. And we put them in that dish, and we allow them to grow, and the neurons start making a little brain, literally. They begin to wire together. And we have a number of laboratories that are trying to do this with a series of mutations to see, because we have a number of mutations that predispose, let's say, to schizophrenia. We don't know exactly how they all act. And one of the great hopes would be there'd be some kind of final common pathway through which these variants would work. Now, I've been talking straight for quite some time. I haven't had a single question. So I'm going to take a breather. Can someone help me out with a bit of discussion? Chip.
Can you distinguish between what mental illnesses seem to be heavily genetically driven and those that don't seem to be heavily driven? You talked a little bit about that, but I think before there are some mental illnesses for which there's no genetic basis. Is that correct?
No. We've not been able to find. There are some disorders for which the genetic influences are quite modest. Let's imagine a simple phobia of spiders. Like that. A lot of it has to do with having an unpleasant encounter with a spider when you were young, but there actually are some genetic predispositions. We certainly know, using these molecular tools, because we now can map the risk of these variants, there are lots of people that don't have any of the risk variants. And there's some that have a high concentration. And we've done big studies. The biggest one actually now is in Finland, of all places. This is a very international kind of process. They've got 25,000 of those people already sequenced, and they can actually pick people out, and they're calling them into laboratories to put them in MRI scanners and others to try to find what might be the main correlates of those individual variants. Okay, please.
And you may be getting to this, but I'm wondering how you account for the variations in severity.
Okay, so the question is, how do you account for the variation in severity? So that is a question that I've actually looked at quite a lot. For most disorders, those that have severe conditions, let's take an example. Early onset is almost always bad. If you have schizophrenia and starts when you're 16, so you haven't had time to develop your personality, develop academic skills, maybe develop some romantic arrangements, getting ill at 16 and 35-year-olds have a lot more to build on. And that we know is strongly related to the genetic risk. And that's true across many diseases. By the way, not just psychiatric disorders. If you have a heart attack at the age of 34, you have something really very highly genetic. Something's really the matter with your cholesterol metabolism for something. But for depression, for schizophrenia, for bipolar illness, in all those cases, the early onset forms are substantially more heritable. And I and others are now trying to translate that question into these molecular markers. So, poor outcome over the course of the illness is influenced by genetics, but not entirely. There are really very important other influences. For example, people with severe mental illness who have good families, who watch after them, help them stay well, get them to doctors. And other families, either the person runs off or they give up. And that can have a big impact on the quality of their survival. Manfred Bleuler, the son of Eugen Bleuler, has written an extraordinary book, which was a 40-year follow-up of a key group of 200 patients with schizophrenia from the 1940s to the 1980s. And he tells those stories about what happens when they're 35 or 40 and how some people seem to kind of wake up and function better over time. And of course, one of the things that he clearly showed is that this is not a dementing illness like Alzheimer's is. Schizophrenia doesn't share that people actually can get quite a bit better at certain times. So there are certainly features of the course that are due to environmental things, but genes do play some role. Yes, please.
So it sounds as if the nature of the research is global, right?
Absolutely.
And it sounds as if your emphasis has been primarily in schizophrenia?
I currently have 2 projects in depression,
Okay.
and I have 1 project in bipolar illness, and a couple in schizophrenia.
So when you look at the global research portfolio and what people are focusing on, what would you say, if you were to break it down in 2 percentages, is a lot of work going into schizophrenia, or a lot going into mood disorder, or a lot into...
Okay. Yeah, I think I could approximate that. Most of us see schizophrenia as the poster child of psychiatric molecular genetics. The sample sizes tend to be bigger. Also, the sample sizes are more carefully done. I think bipolar illness would probably be a close second. It shares a number of features and also shares some genes. So a thing I didn't talk about, remember those 19th century alienists that was noticing that some families that a lot of disorders seem to run in were showing that molecularly, that there's some sharing of genetic risks. And there's even sharing of genetic risks between disorders like schizophrenia and autism, which is a very different condition. We have, and I'm actually a firm believer in what is called the neurodevelopmental spectrum by which, if you think about disorders, on one end is severe intellectual disability, and then you have autism, and then ADHD, and then schizophrenia, and then bipolar illness, and then depression and anxiety disorders. And at each one of those stages, the relationship with intellectual dysfunctioning gets weaker and weaker. By the time you get to depression and anxiety disorders, those folks are normal intellectual functioning. There's really no determined deterioration. And what we see is that the genetics of that changes rather dramatically. That you have more severe, rarer variants in the autism and intellectual disability. And by the time you get to schizophrenia, there are a few such variants, but like 1 out of 500 cases with schizophrenia has that. And they're different in other ways. They have more likely epilepsy, more likely mental retardation, and their eyes are somewhat further apart. They have this syndromal kind of appreciation. So, yes?
How does imaging play into this? MRI, nuclear isotopes, and
Right.
that, can it be used?
Well, when it started,
in
yeah.
sequences?
Yeah. So you should be aware that the history of psychiatry is of getting new technologies and being sure they were going to answer all our questions, and then we get to solution. So when EEGs were developed a long time ago, and were developed by a psychiatrist, said, well, we're going to be, by EEGs, we're going to be able to detect all of schizophrenia. Didn't work out. Then pneumoencephalography developed. And of course, I was working and actively practicing when both first CAT scans and then MRI scans came out. So we are now 35, 40 years after the beginning, MRI scans in particular. It's mostly been a bust. That is, reliable differences between cases. It takes large numbers, and the differences are statistical. So on average, individuals with schizophrenia have mildly larger, what are called lateral ventricles. In our brains, there are pockets where the cerebrospinal fluid collects, and that's normal. But individuals with schizophrenia have somewhat increased sizes, but you can't use that for diagnosis. And there's been efforts to focus down on specific regions, especially the prefrontal cortex, the hippocampus, with the hope that that would show delineations. It hasn't proved very helpful to date. Yes?
Speaking of technology, what are they saying about, or what's being done related to AI and genetics? And do you expect any kind of big advances with that technology?
Every conversation in my field talks about AI, and ChatGPT and I have gotten to be on much better terms than we used to be. It's an excellent research tool. So there's some really technical areas. One of them is the following. Protein structure is very complicated. We now have a program that helps do that, but it's these complex three-dimensional foldings. And we get a mutation, in this linear. So this isoleucine, which is one amino acid, gets changed to valine. What does that do to the structure of the protein? And that's extremely hard to guess because it's complex three-dimensional configuration. And there's all kinds of algorithms been developed, and now AI is competing. And the last thing I read is that they're actually already better predicting the impact of that single amino acid change in the protein on protein function. This old idea now that we're going to take everything we know about schizophrenia, dump it into a computer program, press the button, and go take a short vacation, it's going to have the solution. No way. It's not anything like that. But in these technical areas, it may speed up certain functions. The main problem with AI is that you never know what happens under the hood. So we have this concept of machine learning, which is closely related. You say, all right, I want to get a predictor of schizophrenia. I take the UK Biobank, 1 million people, thousands of pieces of data on it, and put it in and say, predict schizophrenia for me. And you get some algorithm. You can't ever make sense out of what that was doing. That is, computers don't work the way humans do, which is look at causes and come up with them. So there are some limitations in that area. But it's a good question. Please.
Do you use or recommend gene site testing, or do you see a correlation in families that are grouped together that their treatment plan works well for the same?
Okay. So let me take a little bit of a step back. So polygenic risk scores. When you have a genome-wide association study, and let's say for schizophrenia you have 400 variants that are significantly different between cases and controls. You can take, in imagination, you're taking all the control DNA and lining it up with all the schizophrenia DNA, and then for every single base pair, you're looking at the average difference and just summing that up across the 3 billion bases. That's a polygenic risk score. It's sort of a summary. It's been an important research tool. So now if you look at a polygenic risk score, if you're in the upper 5% of people with a polygenic risk score, you have a risk of schizophrenia of about 4%, almost exactly like that of a sibling of a patient with someone with schizophrenia. So it's not 50%. We can't come close to doing that. And that, by the way, would be the figure if you had an identical twin with schizophrenia. Your chances of developing schizophrenia would be about 1 in 2. Are people able to hear me clearly? Okay, they're still nodding. All right, thank you. So it's certainly a research tool, and we can ask fascinating questions. So, for example, there have been in big population samples, what is a 7 or 8 or 9-year-old like that has a high risk for bipolar disorder? And it turns out that they're a little impulsive, they're more moody, they have a little bit of ADHD symptoms. We couldn't have answered those kind of questions ahead of time. That what might be the subtle manifestations of a higher risk for this disorder. So it's an important research tool. There is a pressure from capitalistic enterprises to start selling this on the web. Now, we will tell you your genetic potential for intellectual, into educational attainment, into risk for alcoholism, risk for schizophrenia, risk for diabetes. And even of greater ethical concern to us is that people are now offering these services to women who are doing IVF. So if you have 8 zygotes and you want to go and test them to get the one who has the higher IQ, the lower risk of schizophrenia, you can kind of go on by that. We have an ethical board in our committee. Their comment is this is not ready for prime time. First, you're comparing siblings already different. The prediction isn't that good, and the potential misuse is pretty scary. So it's a situation where we're going to have designer babies, and it's not. You wouldn't be surprised at who are the ones that are pushing these companies. They're often the mega rich, high-tech folks that are investing in this because they think there's going to be some big call for this going on. So we're watching that carefully. It's not, Congress has not done anything except the outgoing head of NIH, Francis Collins, did get a law passed that insurance companies cannot alter your policies on the basis of your DNA sequences. That's very important. So that protection is there, but nobody in the government has yet tried to figure out how to regulate these sorts of issues. And I personally am really quite concerned about that. It's kind of like the metaphor. There are times when human knowledge runs ahead of human wisdom, and you can really start getting into situations of misuse. Good. So we were at the point of discussing what are the new technologies that are available for molecular studies, and we began to talk about transplantation of the genes into rodents, into primates, to try to look at effects. So what we have been able to find is that when you add the mutations that predispose to schizophrenia or bipolar illness in rodents, you get subtle abnormalities of social behavior that you can measure. And there is some evidence that there is convergence between the different mutations. The most dramatic work has been done in China putting these genes into rhesus macaque monkeys. Rhesus macaque are old world monkeys. They have very complex social behaviors, and we were exposed to one of the high risk variants for autism, which is called SHANK1. And the screen this was put into a male rhesus who is now 3 or 4 years old. And if you're used to watching the rhesus macaques in a zoo, these males run around, they tumble, they fight, they pitch things at one another. It's very active, very athletic, and very social. These males often have been raised together. And we were shown videos of these young rhesus macaque monkeys containing the SHANK3 gene, and they were kind of in a corner. They were trying to participate, but they weren't skilled. They had aggression pointed toward them because they seemed to be so socially dysfunctional. So you can't make conclusions on that sense, but do we now have the capacity, and you can talk about the ethics of that, to be able to now take these variants, put them into primate brains, which resemble human brains in important ways, and carefully study what the possible impact might be. Now, I want to also talk about this technology of actually making small brains. I talked to you initially about the concept of brain in a dish, in which it's in a two-dimensional structure. But we now have the capacity, by putting them in these swirling, warm, liquid baths full of nutrients, to allow them to develop in 3 dimensions. And you can grow them for as long as 9 months, and these neurons start forming into what are little brains. And we can talk about the ethics of this at some point if you want. They don't certainly experience consciousness, but you look at the slices, and well, here's a little bit of retina here, and here's a little bit of frontal cortex there, and they're starting to wire up. The question now that we have, among the various variants we're discovering, will we find this concept of equifinality, or a final common pathway? Could it be that we have 5, 6, 8, 10 variants, but at a bottom part, most of them or all of them come together in a single pathway? Or is it a more pessimistic or complex picture that these variants look very, very different from one another? So one of the key issues as we have begun to assess these variants is how coherent are these various groups of biological changes. So one model is for those of you that own a shotgun, you take your shotgun out to the back of the barn and you just take one of these giant maps that you used to see in biochemistry labs, which have every single protein synthesis pathway of lipids of the whole human in this big path. And you take your shotgun and kabloom, you just blow a bunch of holes in it, and there's no rhyme nor reason of those occurring. That would be a pessimistic outcome. And what that would mean is that mostly anything bad you do to brain functioning will predispose to psychiatric illness, but we're not getting our hands around one final common pathway. We already know that that's not true. So even at the preliminary areas we have now, we know that there are variants, for example, that impact on an important neurotransmitter, glutamate. So we have 3 or 4 in that area, clearly cannot occur by random chance. But then we have other sets of genes that are involved in DNA synthesis and control. So clearly the idea that as we get the variants, they're all going to map onto one another, we can look, think us climbing up that rope to the point that we're going to have a single solution in biological pathway is not occurring. But there may be a small number. And that is where the active developments are. So one of the things we're doing is building from dozens and dozens of human mutations, these little brain variants, put them in vats for 9 months, and then look at the brain patterns. And in some cases, we seem to be seeing some common patterns in the way those genes are beginning to wire together. So that would be sort of very cutting edge kind of current approaches. Bipolar illness has been, so let me take a step back, and what I want to do now, since we have enough time, is just to describe to you what a modern field study in psychiatric genetics is like, to give you a kind of on-the-ground approach. So the study I'm going to talk about is called ABIGNet. It's Asian Bipolar Genetic Network. 4 years ago, when the National Institutes of Health realized that if you took all of the human genetic information that was available and you looked at how well represented it was across human ethnic groups, 98% of that data was in white Caucasians. And we had very little data on people of African extraction or South Asian or East Asian extraction. And that wasn't right. It wasn't right ethically, and it wasn't right scientifically, because in fact we contain only a small part of the human diversity, and there would be other variants sitting in other racial groups. So they issued large grants. Typical NIH grant now is about $2.5 million, which is not nearly enough to run one of these large studies. And they issued these mechanisms that gave grant sizes of $12 to $15 million, which you can really do with. And I was able to be successful in 2 of those applications. I will explain one of them now, but I can tell you the second story. So ABIGNet was designed to try to assess bipolar illness. And if you looked at the map of what we knew about bipolar illness, the vast majority was in Europeans, but some studies have been done in Latin American populations. So the area that there really been no progress in was both South and East Asia. So a team of 4 of us assembled together, One of the investigators at the Broad Institute and Stanley Center. For those of you not familiar, the Broad Institute is probably the preeminent institute in molecular genetics in general in the United States. They played a key role in sequencing the human genome. They're big players, and it's always impressive when I go up there. And then Johns Hopkins and VCU. And one of my graduate students who is now working in Taiwan. So the four of us were a team to try to put together a grant proposal. And we first begin to ask, what kind of disorder are we going to study? We agreed on bipolar illness. Do we want to take a narrow view or a broad view? And I argued strongly for the narrow view, and I won. It was a pretty intense argument. And then how are we going to assemble both East Asian and South Asian countries? China is now off the map. One is not, as a US researcher, able to collaborate with them. It's a long story. I don't think we need to go into the details. Japan is a place you can work, but it's extremely expensive. And the sample size is fairly small. So through extensive selection process negotiation, we decided to work in Taiwan, where ProShow is. We decided to work in South Korea, where we also have a depression project. We decided to work in Vietnam, which is really transitioning to a second world, and they were extremely enthusiastic. So for a place like Vietnam to have this amount of infusion of money that would allow them to establish collaborative networks, and then Singapore, which was mostly for symbolic reasons because it's pretty small, and then India and Pakistan. So we set out trying to develop interview protocols, figuring out how many languages we had to translate them, what kind of questions we wanted to ask, and we got the grant funded. It took a long time. Total budget was a little over $15 million, and we proposed that we were going to try to collect 27,000 cases of interviewed bipolar disorder and 20,000 controls, and they would be distributed across these countries. It was my job to try to figure out how to train all these interviewers. So at odd hours, because of the link, I could either talk with colleagues in Asia in the morning, like 6 or 7 a.m., or 8 or 9 at night, because in the middle of our day, they're all asleep. And I would have my Zoom screen filled with 20 or 30 people as I'd be talking about the various interview strategies. We talk about the cases. We do practice interviews together. I would train the individuals. Some would fly over, or I would fly there to visit them. And we established, with help here, a quality control center at VCU trying to monitor those collections. The group at the Broad dealt with the DNA. We decided to use the most advanced genotyping now. It's called the blended genome product that allows us to look at the whole genome but gets us that exomic variance very well. And we now meet as a group every 2 weeks. We've probably had about 150 meetings. And we also run every other week what we call the site phenotype leaders, in which we have representatives from every one of the countries. We spend an hour together. We begin by them telling us how their data collection is going. We then talk about quality control issues. Are they having some problems with interviewers there? We then talk about some science, because one of the things that we're doing, and I'll give a bit story of that, is trying to encourage their own scientific work. And then we take a case and decide one together. So our last case was a person who developed bipolar illness after coming down with multiple sclerosis. And should we include such a person in our study? The case before that was someone who had their first onset of mania after using a lot of stimulant medication, which can also cause mania. And for me, the experience of having people from these diverse cultures, psychiatrists, talking together, we all know one another, we're on a first-name basis, forming these teams now scattered across the world. Our sample collection is now at about 12,000 cases. And we're going to aim for 25. It's just remarkable. I've gotten to fly to India twice, and I've been put on this one hospital, one-day tour. Getting up in the morning, getting to the airport, flying there, going out, meeting at the hotel, meeting with the head of the hospital, going and meeting with the psychiatrist. I usually get an hour or two where I can meet with the individual raters. And in India, fortunately, they're all English fluent. We'd interview a patient together. We'd talk about the interviews. We'd go out to eat dinner. We'd go to bed, wake up, and repeat across 10 different sites across 2 trips. The scene in India, we think an outpatient clinic in Delhi, in New Delhi, people would start arriving at 6 a.m. in the morning. And by the time the offices opened at 8, there'd be about 1,500 people waiting to be seen. They'd just show up in the morning, they wouldn't have an appointment, and they line up. And a junior psychiatrist would have literally a line of people to be seen. They see them for 15 minutes. They write these things in detailed English, quite high quality psychiatry. But the magnitude of the population and the variety of the population. They also have a practice in all their psychiatric hospitals. You can't be admitted without a family member, which is almost always the mother. So you would see samples at a schizophrenia ward with these young people, and there's the mother. She's helping with the baby, washing the clothes, keeping the person. Right? And what they say is, oh, the mother, much less agitation. People are less upset. We have someone to help us and calm down. We have discussions about religious delusions. So in mania in particular, the delusion that you are God, the Son of God, Moses, it's very common, these so-called grandiose religious delusions. I was a religious studies major in college, so I know something about Hindu mythology, but there's a whole lot of gods there. And the sort of question is, how are we going to interpret religious delusions in those contexts? So it's been a very exciting process. We're about a little bit more than halfway through our grant, about halfway through our deadline. We think we'll get close to meeting that deadline, but the amount of hours and work is just extraordinary. So that's what a modern study is like. Okay, stop for some questions. Yep.
So I'm not able to hear everything. So
Okay.
I'm just wondering if you can kind of summarize maybe the aspects of your research that can most help us as we're supporting our loved ones with mental illness.
All right, I have a big research portfolio. Certainly a good deal of my effort now is to find populations where we have good collaborators to collect large numbers of samples to assess them quite carefully psychiatrically, and that's part of my job. And I train and then collect DNA samples, take them back to the lab, and try to understand where those genetic variants are, and then with our neuroscientist colleagues, not me directly, track back to understand the basis of that biology. So that's one thing I do. I have a little specialization in that I don't think that the disorders as we understand them now are unitary concepts. And I am convinced that we have to peel back different subtypes of that group by doing careful clinical evaluations. For example, in bipolar illness, about 20% of patients never have depressive episodes. They only have manic episodes. There's got to be some difference there. 30 or 40% of bipolar cases develop quite substantial psychotic symptoms. I am the king of the world, I control the weather, I am the best friends of very famous people. I walk down the street and all women, are very attracted to me. I can fly. That's a very dangerous delusion, of course, because people jump off of buildings. What's the difference genetically between those that are so-called sweet manics that just have euphoria but don't develop psychosis? We're looking very carefully at the responses. The symptoms of mania differ between different groups. So my part of my job is that I've got this clunk of symptoms that are moderately homogeneous, but not entirely. And I want to get my hands and start pulling it apart with a hope that those will be more unified genetic entities. In another part of my research, I work with registries in Sweden that don't involve DNA at all. And there I'm interested in asking questions like, how can I look at differences across different kinds of major depression? And it turns out that I can identify from the age of onset, from the level of recurrence, quite different genetic risk patterns, all of whom look like they have major depression. I'm doing the same with schizophrenia, and so I'm using the Swedish registries as a natural laboratory. Swedes know almost everything about you, where you live, the quality of educational attainment, how you did in the army. So it's not got molecules in it, but it allows me to ask finer questions about subtypes, and I can take those lessons and then apply them into the molecular projects. So that's a lot of what I do. Please.
Oh, you mentioned with the shank gene, it was
Yes.
specifically in male monkeys. Would the results have differed at all with females? And sort of going off of that, have you found any correlation between sex and mental disorders in general?
Okay, so let me ask the first one. First, I didn't make the decisions about who got the variant in SHANK3, done by specialists. We know that autism has a sex ratio, especially in the early onset form, of 2 to 3 to 1. So it's considerably more common in males. I think it also they might have thought that testing the social behavior of male rhesus, they're much more active and running around, might have been easier, but I don't really know the rationality. It's, I think, something like $50,000 for one rhesus monkey to do this. So it's something you're doing in very small numbers. Now, your second question, there is very few psychiatric disorders that do not have a prevalence that's substantially greater in one sex than the other. Major depression, anxiety disorders, many of the personality disorders are much more prevalent in women. Bipolar illness, only slightly more prevalent in women. Schizophrenia, somewhat more in males. Alcoholism, drug abuse, substantially more predisposed to males. The fascinating part is if you sum up the total, it's not very different between the sexes, but the manifestations of them differ substantially. And there are many, many theories about why that is. We could maybe talk about that afterwards. So, Bernadette, you had a question?
So all of this work, obviously, you're learning as you're going along.
Absolutely.
So it has to then going into the clinic, like the medication usage that happens there. Has there been research that's shown like where one medication is going to be better used in this, I don't know if you'd say genome or whatever, versus like another? Like, have we gotten to that part? Is that?
So there's a field that studies that. It's called pharmacogenomics. Not surprising. It has been rather disappointing. First, as you hopefully have learned, modern genomics of psychiatric disorders you can only do with really large samples. And the only way that you can really understand when a drug is really having an effect is a randomized double-blind controlled trial. And big ones are about 300 people. So in depression, if you averaged across all the SSRI controlled trials, the placebo response rate is about 35%, and the SSRI response rate is about 45%. It's clearly bigger, but a lot of people just get better on their own. And so there is much effort to go into biobanks and try to untangle what's going on from who changes drugs and who's others, but I'm quite pessimistic about that because it's very confounded. Interestingly also, in the studies we've done, we've been much more successful at predicting who gets bad side effects. So it turns out one of the really negative effects of the so-called new antipsychotics, the risperidol, olanzapine, is they really increase your weight and they give you type 2 diabetes. And that turns out to be genetically influenced. So at some point we might be in a position of getting a genetic screen that will at least tell you not what's going to be more effective, but which ones are going to have bad side effects, because those look like they're more heritable so far. Yes?
I was interested in one of the cases you were talking about with the later onset of bipolar disorder with someone who had been prescribed, I guess, stimulants for a period of time. Have you seen, I guess, any relation to that with folks who are, say, using like methamphetamines or cocaine, like any later onset bipolar? I'm curious if there's any.
There is a clinical literature that stimulants in general, including all those compounds, as well as the dissociative, the ketamine and others, that they predispose acutely to states that can resemble mania. Most of those cases, when you follow them up, are clearly not bipolar illness. They get better. I studied that not for drug-induced bipolar illness, but for drug-induced schizophrenia, again in Sweden. And we really got quite definitive pictures. So roughly, if you look at the people who develop psychosis while they are taking stimulant medicines, they do have an elevated genetic risk for schizophrenia, so they have some propensity. But then you follow them up over long term, and some of them go on to develop schizophrenia and others get better. And it turned out those that go on to develop schizophrenia have genetic risks that are indistinguishable from regular individuals, which predicts that probably in that situation, the drug is more bringing out the vulnerability that was there. And if someone gets, use the vernacular, crazy, and you wait 6 months, most of those get better and back to normal, if they don't use drugs again, of course, that those probably are usually people not at high elevated risk. Cannabis was a bit worse, and alcohol was the most benign. Few people get psychotic symptoms. And you may know, because of the legalization of cannabis, there's a huge scientific row in the literature about whether that's a good idea, how much might you be increasing risk for schizophrenia. I could give a 15-minute talk just on that, but that's really controversial now.
Can you follow up on that issue?
Okay.
Marijuana and schizophrenia.
All right, so the basic finding is we use a method called a co-sibling control design. So if you, let's go to Sweden, and you find an individual who has gotten a registration in a hospital for cannabis abuse, they have an increased risk of schizophrenia about 9-fold higher, really large. And then you go back and you compare them not with the general population, but with their sibling who didn't have any cannabis problems. That risk goes from 9 down to about 3.5. And that shows you that there's something about sharing genes, sharing rearing environment. And if you just look at that 9 figure and you're going to make these epidemiologic predictions, you're way overestimating the effect of cannabis there. So it's a way of moving from the classic correlation to a better idea of what's causing stuff. And one of the issues with epidemiology, I just count things and I say, oh, that's correlated with that. That doesn't allow you to plan intervention programs. It's the old, drinking a lot of orange juice is related to a low risk of heart disease. You think that we just drink more. That's not, of course, true. People who drink a lot of orange juice don't tend to smoke. They tend to exercise a lot. They keep better control of their blood pressure. And so you have to be very careful about those kinds of inferences. So the conclusion of that is cannabis is bad, but not nearly as bad as people are estimating. Yes?
With the behaviors that are escalating in school systems, are there any local things that you're researching with the children currently and increased autism, ADHD, or
Okay, so the answer is, what am I doing vis-a-vis school children? The answer to that is no. I am a trained adult psychiatrist, and I have occasionally used in some of my models work with autism or ADHD, but I've not tried to run primary studies there. Those are both quite crowded fields. I could spend a minute, if we have, to just talk about some of the advances in autism. That's very, very interesting. So, Elise Robinson has worked at the Stanley Center for about 8 or 10 years, and she is the first person who observed, let's look at a group of autistic children as a function of their IQ, and then look at their risk for these rare kinds of variants that I said you need to detect by doing exome sequencing. Those are changes that are doing quite bad things to your brain functioning. And what she found is that all of those individuals with that kind of autism had quite low IQs. They often with a kind of flapping and headbanging form of really severe autism that you might be familiar with. And they had very high burdens of those kinds of rare variants. Once you got to IQs of about 100, that fell away entirely. And if I will give you, pardon my expression, the Dustin Hoffman kind of autism, these Asperger quite high functioning, but it won't look you in the eye, to say I'm on the spectrum. That group has none of those variants. They just have these polygenes that predispose. So that's an example where the genetic architecture of autism changes dramatically as a function of this IQ measure. ADHD is more complicated. It doesn't work quite that way. ADHD is unfortunately related to large subsequent risks of drug and alcohol abuse. And that partly has to do with poor school performance and others. So that's about the summary of knowledge I'd have in that area. Yes, please.
Has there been any evidence for a role of epigenetics in the suppression or expression of these genes?
Okay. Usually I get asked that question once or twice. So to be familiar, DNA is complicated enough, but we have to do things like silencing our genes. So an egg has a potential to be everything. How do you get from an egg cell to a liver cell, to a muscle cell, to a brain cell, to a pancreas cell? And what you need to do is lay down these methylation marks to silence whole hunks of genes so your liver cell doesn't all of a sudden start doing things your pancreas cells are supposed to do. And those last for a long time. And it's also known that methylation in the brain is at least partially involved in memory. Memory is a very mysterious thing. How does our human brain carry all these? But one of the ways it does it is actually changes the methylation pattern of genes. And we know that stress impacts on methylation. So, for example, I was involved in a study. I had done the work on our twin sample in Virginia of people reporting childhood sexual abuse, which in severe forms in Virginia, like many other places, occurs about in 3% of women. And it substantially increases risk for subsequent illness. I had colleagues at the institute I work with that asked me whether we could try to work with that group to obtain blood samples to look at whether methylation patterns were different, particularly in the genes involved in the stress response and those that had the severe stressor. Of course, we wrote the twins, we got informed consent, and lo and behold, they found that. So it's not the inherited kind of variation. So, and it's changeable over time. So, you now, and you after you have a bad cold, or you're 20 years older, or you're taking extra medicine, those methylation patterns are going to shift around. So my general comment of this, I've got enough trouble just figuring out what DNA is doing that is basically stable in your body. I can let other people worry about the methylation, which is, it's a more dynamic aspect of the genome. But it's an appropriate question. Yes, please.
Can I come closer? Because I'm having a hard time hearing, and I want to make sure people can hear what I have to say.
Okay, please step up.
You might kind of have somebody similar to this. I have a son that has schizophrenia, and it's kind of rough.
I know.
And it's tough, challenging. The biggest aspect that I have a problem with is he feels like he's in this wonderful Anastasia world of enlightenment and energy, and the drugs that he takes puts him down here. So he doesn't like to take drugs. So I want to know how to help my loved one, because I love him to death, but he's been on this roller coaster ride for years, 10 years, battling schizophrenia. So I know environment has an influence, my genetics has an influence, but as a psychiatrist researching this, like, what is the best way to help someone understand that, yes, you can make your decisions, but there are consequences associated to this, and to see outside the boundaries of themselves? Or is it impossible? Am I on an impossible journey with everyone? I mean, I would love to say that I'm a very spiritual person, and I feel like there's hope for many of us. And we need that. We need to know how to help our loved ones. And I came here and I texted you like a question or two because I'm frustrated with the system. So is there any kind of encouragement that you can give us in this room? I know there's someone just like me. There's many of us here that might be kind of going through that.
It's a very good question. I wish I had answers that I could deliver for you now, but I don't. The science is making progress, but we're not close to developing treatments. One of the unfortunate features of our field is that every single medicine that has been found for treating schizophrenia has been found by accident. It all started out with chlorpromazine, Thorazine, and that they thought it was an antihistamine. And some French guy gave it to a bunch of people who were mentally ill and noticed that they kind of calmed down. And every modification we made from that is not because we understand etiology.
And he loves his stimulants. I'll tell you, he loves dabbling into things,
Yep.
And he feels like pot is the answer. It puts him into a settlement of where he can calm down after that mania. But then it has a negative consequence to it that he can't see, and he's not exposed to it. It's just, I wish I had all the answers, but I feel like the system's broken. There's a broken system outside our world to even get the support and help that we need.
That is a different question. I absolutely agree with you. The way we deliver mental health in the United States, on average, is much poorer than in other countries in Europe in terms of providing care.
Just gear us in the right direction of contacts that we can develop a roadmap. Because I feel like there is no roadmap. It's just like we're on this journey and it's like a roller coaster. So I'd like to get to the end of it and come through the maze just so that the loved one that I'm trying to support has it, because everybody else has broken away. I'm like the only survivor, and that's unfortunate. It's like for some of us here, we're the only ones with that mentally ill person. And I don't want to fail. I'm a fighter, I'm a little tiger, and I want to win this battle.
So my dream, and whether it will be in time for your son, is to use the findings we're having in genetics to identify mechanisms and then develop drugs that are specific for those mechanisms with much more benign side effect protocol than we have now. So that your son wouldn't get fat or wouldn't get drowsy and would have just a specific impact on his delusional thinking, and that would be all it would do. But that's going to be years. No, it's not there. And it's not for lack of trying.
He's working on it, Donna.
Yeah.
He's working on it. Yeah. Well,
But you're right to be frustrated. If I had a child.
Person, let me tell you, because I figured everybody needs to hear it, and everybody's probably in the same category. But thank you. I appreciate all your.
It's a
your
good
research,
message, and I think about
I
these.
hope we come up to the end and we get to the puzzle. you know, in our lifetimes, but thank you.
I do think of these things every day.
Yeah.
So, yeah. Okay. So I think it's probably about time to wrap up. Any final questions? I'll stay for a few minutes afterwards. You've been a very good audience. I want to thank the MCV Foundation and VAMI for putting this on. And of course, Chip Lacey, whose original idea this was, stimulation. So it's been a meaningful experience for me connecting up. And I think that last message is a good one to remember. That is that we're making progress, but it's not soon enough. And people are working hard. I work with, of course, lots of people who don't have psychiatric experience. And for them, this is more of a theoretical issue. And I'm the one who often makes the point in these meetings. There are real people out there. And one of the reasons why I accept every invitation I get from VAMI or NAMI is because it gives me contact. Because in some ways, you are my natural constituency. You are what I'm trying to bring through. And what will get on my gravestone or not, I don't know, but we're working hard. So thank you.